Coming to the acetylcholine-rich receptors, it is of two forms, adult or mature or junctional receptors, fetal or extrajunctional receptors. Adult receptors are formed with 2 alpha, 1 beta, 1 epsilon and 1 delta, whereas the fetal receptors are formed with 2 alpha, 1 beta, 1 gamma and then 1 delta subunits. The fetal acetylcholine receptors, they can be developed in these conditions like prolonged immobilization, burns, sepsis, neuromuscular disorders, upper motor neuron lesions, lower motor neuron lesions. The fetal acetylcholine receptors are more sensitive to acetylcholine. Coming to the mechanism of action of the neuromuscular junction, when the action potential reaches the nerve terminal, it opens the voltage-gated calcium channels. When the voltage-gated calcium channels are opened, there will be a heavy inflow of calcium ions. It exerts attractive forces on the vesicles clustered in the zone 1 and they fuse with the neural membrane and simultaneously release of acetylcholine molecules into the synaptic cleft. Acetylcholine released in uniform sized packets called as quanta, which contains 10,000 molecules of acetylcholine. These quanta, the number of quanta is directly proportional to the intracellular calcium channels. Its application we will discuss in further thing. When the muscle is stimulated at higher frequency, zone 1 packets will go to the zone 2 packets will go to the zone 1 and with more acetylcholine available for the impulse transmission. So at higher frequency, calcium enters the presynaptic terminal during each cycle. So during this high frequency period, there will be no time for the calcium to go back to the extracellular fluid. So calcium is concentrated there, high concentration of calcium channel. So coming to this implication, the number of quanta are directly proportional to the intracellular calcium. So with high concentration of calcium, there will be more number of quanta that are released and which causes strong muscle contraction, which is used in the neuromuscular monitoring. And this is the acetylcholine synthesis in the nerve terminal. In ECF and mitochondria, the choline and acetyl-CoA are combined with an enzyme acetyltransferase. Acetylcholine is formed and it is packed in a quanta which are strategically positioned for the release.